Okay, welcome back. In this lecture, we're going to get into talking about the gene structure of MHC. What is it about the genetics or the genetic information that encodes MHC molecules that allows them to contribute to the overall diversity of our antigen recognition repertoire? So as we saw with immunoglobulins and T-cell receptors, we got a huge diversity in the different types of antigen receptors that we could make by the recombination of different gene segments. So the genetics of MHC also contribute to diversity, but they do so in a completely different way. We don't recombine gene segments to make different types of MHC, but as we'll see, there are other ways that we encode an ability to make slightly different versions of MHC on all of our cells to make sure that we can capture a broad variety of different antigen types in order to present them to lymphocytes. So two major ideas here in the diversity of MHC are that MHC genes are both polygenic and highly polymorphic. So what does this mean? Well, a polymorphic gene is one that has multiple alleles. So recall that for any gene, we inherit one copy of it from our male parent and one copy of it from our female parent. And so we have two chromosomes, one from each parent. And many genes have different versions of themselves that appear in different people with different frequencies. We call those alleles. And so, for example, for this gene, this particular cell happens to be heterozygous. It has one allele on one chromosome and another allele on the other chromosome. And so when multiple alleles occur for a gene, we call that polymorphism. So for MHC, certainly, there are many polymorphisms in MHC. There are, as you'll see, many hundreds and thousands of different versions of each of the MHC genes. And so for that reason, there's a lot of diversity in the potential MHCs that you might inherit from either parent. The other important idea here is that MHC genes are highly polygenic. So in addition to getting diversity from polymorphisms, we actually literally just encode multiple distinct genes of the same, that encode the same protein, basically. So for example, for MHC class 1, we have 3 different genes, which means we can make 3 different MHC class 1 molecules. And you can see this here. In this example, though, we have polygyny, but not polymorphism. So here we have 3 genes encoding MHC, but this cell is homozygous for each of the 3 genes here. So in total, then, we're going to make 3 different versions of MHC class 1 on the surface of this cell. Well, if you combine these ideas, though, polymorphism and polygyny, you could have different alleles on each of the 3 different genes. And so in this case now, we can make up to 6 different versions of MHC class 1 on the surface of this cell. Now, an important distinction to make here is that in the case of immunoglobulins and T-cell receptors, each of those molecules was recognizing one single antigen. That's important for lymphocytes, but we don't need MHC to be quite that restrictive. So MHC molecules can bind to a broad variety of antigens. They can capture a lot of different molecular shapes, basically, of the peptide fragments that they bind to. But remember that the peptide binding groove is itself a protein, and it has a particular shape. And the shape of any one MHC molecule is just not able to accommodate all of the possible peptide shapes that it might need to bind to in order to present. And so for that reason, even though individual MHC molecules can bind to a large variety of different antigens, we need sort of a collection of slightly different MHCs on the surface of our cell so that collectively, if you sort of picture a Venn diagram, if we have 6 different versions of MHC class 1 on the surface of our cell, then that's going to cover most of all of the different possible antigen types that we might need. Some of those antigens might be only presented by some of the MHCs, but collectively, we're covered across the entire spectrum of all of the possible antigens that we might see. So that's made possible because MHC genes again, are both polymorphic and polygenic. So for example, for MHC class 1, the combination of those two ideas means that we can make basically 6 slightly different versions of MHC class 1 on the surface of our cells. So let's look at the actual structure of the MHC genes in a little bit more detail. So in humans, all of the MHC genes are encoded on a single chromosome, and so they're all right close together. So this is also a source of distinction from lymphocyte receptors, which were on 3 different chromosomes. Here, all of the MHC components are found on the same chromosome. That region of the chromosome in humans we call the HLA complex, or human leukocyte antigen complex. And so frequently when we're talking about human MHC, we use the designation HLA, but that's what we're talking about. Within the HLA locus, within humans, there are 3 distinct regions which encode the different classes of MHC. So first, there is the MHC class 2 region, then a region that we haven't talked about called MHC class 3. The only thing you really need to know about MHC class 3 is that it encodes sort of accessory immune proteins that do other things related to the immune system. So you can see here, for example, TNF-alpha, a cytokine that we've talked about, C-reactive proteins that are involved in the innate immune response. These are immune molecules, but they're not involved with antigen presentation. So we won't say anything else about MHC class 3 in this lecture. But after that, we have MHC class 1, which of course we should talk about here. So we'll focus within the HLA locus, within the HLA complex, on the genes that belong to MHC class 2 and MHC class 1. What you can see here for both MHC class 2 and MHC class 1 is that there's actually 3 distinct coding regions within each. So this is the idea of polygyny for each of these. So we encode 3 different MHC class 2 genes, which means we can encode 3 different versions of MHC class 2, basically, for each of the chains. And same thing for MHC class 1. We encode 3 different versions of MHC class 1. These designations, it gets to be a little bit of alphabet soup, so we'll do our best to go slowly through this. We'll start with MHC class 1 just because it's the simplest. Each version of MHC class 1 is identified by a different letter. In this case, it's just A, B, and C. They're in a different order in the genome, but that doesn't matter. So when we talk about all of the different versions of MHC class 1 that we express in our cells, we can refer to them as HLA-A, B, and C. So HLA-A, B, and C are all different isoforms of MHC class 1. For MHC class 2, it's slightly more complicated. For one, remember that whereas MHC class 1 is encoded by a single gene, which encodes the alpha chain, beta-2 microglobulin is somewhere else. So we only need one gene to make MHC class 1 because it only has a single chain. Remember that MHC class 2 has both an alpha and a beta chain. So we actually have 6 different gene products here. We have 3 regions which encode 3 different forms of MHC class 2, like I said. We call those DP, DQ, and DR. But within these 3 regions, we have different genes that encode the alpha chain for DP or the beta chain for DP. And so DP alpha chains come together with DP beta chains to make one DP MHC class 2 molecule. And the same thing for DQ and DR. So again, when we're talking about MHC class 2, you might see the designations HLA-DP, HLA-DQ, and HLA-DR. We're talking about MHC class 2, but we're talking about the specific isoforms that are encoded by these distinct regions within the genome. So this is human. So if we're talking about HLA, we're talking about human. But a lot of the foundational research in MHC comes actually from mouse studies. And so particularly those of you who are interested in research in immunology in some way, you really should be familiar with the parallel gene structure of MHC in mouse. And so it looks a little bit different in mouse. In mouse, instead of HLA, we call it the H2 complex. And so mouse MHC genes are designated by H2. But you can see the same thing here. We have some MHC1 genes, we have MHC2 genes, and then also an MHC class 3 locus, which encodes similar things as it does in humans. But focusing on the 2 classes that present antigen, you see that the MHC class 1 genes are a little bit separated from each other on the chromosome, but again, that's just where they are. They still have the same functions. And there are 3 isoforms of MHC class 1 in mice. There is what we call H2K, H2D, and H2L. So this is analogous to A, B, and C in the human MHC class 1 locus. Instead, mice have K, D, and L. And so when we're talking about H2K, H2D, H2L, we're talking about mouse MHC class 1. For MHC class 2, it's also a similar structure, although a major difference is that mice only encode two forms of MHC class 2, and we call those IA and IE. So whereas in humans we have DP, DQ, and DR, in mice we have IA and IE. So relatively fewer than combinations of MHC class 2 possible for mice compared to humans. Same thing though. These gene regions encode both alpha and beta chains, which need to come together to make the functional MHC class 2 molecule. Okay, so let's look then in a little bit more sort of genetic detail, starting with the mouse chromosome. So all the H2 complex, the mouse MHC complex, is located on mouse chromosome 17. And so we can see all of the genes laid out here. Remember that the class 1 genes are separated on the chromosome, and the class 2 genes and the class 3 genes come in between. But if we look at all of the genes on the chromosome, we see our three class 1 isoforms, K, D, and L, where we would expect them to be. And we see the MHC class 2 genes that we expect. So we see the IA beta chain, the IA alpha chain, the IE beta chain, and the IE alpha chain. So these are the antigen-presenting MHC class 2 molecules. But actually within the MHC class 2 locus, we have a number of other proteins that hopefully you might recognize. These include O alpha and O beta. So these belong to remember DO, which negatively regulates antigen loading onto MHC class 2. In mice, we call that H2O, but same thing. There's also TAP, which is important for loading antigen onto MHC class 1. The LMP molecules are part of the immunoproteasome. So the idea that's important here is that in addition to encoding the actual antigen-presenting molecules, the H2 and the HLA loci also encode a number of other immunologic proteins that are important for antigen presentation. All of these proteins are kind of packed right together in the genome. And so we find them right next to the MHC class 1 and MHC class 2 molecules. So that's the structure of the mouse genome, or the mouse H2 locus. In humans, all of the HLA genes are on chromosome 6. So if we look at the structure of the HLA complex, we see the same thing. We start with the MHC class 2 genes. We see the beta and the alpha chains of DP, the beta and alpha chains of DQ, and so on for DR here. Within the MHC class 2 region, we also see DO, we see DM, which we saw before as a positive regulator of antigen loading onto MHC class 2. We see TAP, we see immunoproteasome subunits, the same as we saw in the mouse locus. Skipping over class 3, we go to class 1. Now we see the three isoforms of human MHC class 1, HLA A, B, and C. And then there's some additional HLA genes in the class 1 locus, which are not involved with traditional antigen presentation to CD8 T cells. They have other immunologic functions that involve primarily natural killer cells, which we're not going to get into in this course, but important to know that there are other MHC class 1 genes, but they're not involved in the classical function of MHC class 1, which is to present antigen to CD8 T cells. That is restricted to A, B, and C. So yeah, there's a little bit of key to the sort of extra proteins down here that I mentioned before, the ones that are involved with the proteasome or the transport of antigen peptides into the ER, the things that we've talked about before. Okay, so we see all the genes here and where they are in the chromosome. Let's connect back to the original topic that we introduced in this lecture, though, which is that here we see very nicely the polygenic nature of MHC genes. So we see, for example, we see that there are 3 genes that all encode MHC class 1, and so we can make 3 different isoforms of MHC class 1 because of that. Same thing with MHC class 2. We have 3 versions of each chain of MHC class 2. But all of these MHC molecules are also extremely, well, not all of them, but most of them are highly polymorphic as well. And so here, this gives you a sort of breakdown of the relative amount of polymorphism that we see in each of the major MHC isotypes. So remember that HLA A, B, and C are the main class 1 presenters to CD8 T cells. These are ones we typically are talking about. The alpha chain, so remember that we encode these with an alpha chain, for all 3 of them are extremely polymorphic. So we have many different possible alleles of MHC class 1. And so that means that each individual person that you meet is likely to have a really diverse and random combination of a lot of different possible MHC class 1 alleles. In contrast, HLA E, F, and G, which I said are not involved with traditional antigen presentation, they are relatively less polymorphic. So you can see HLA F is actually monomorphic. There's actually no polymorphism for that one. And there's only like a couple or a few versions of HLA E and G. And this makes sense because they're not involved with antigens, so we don't need a lot of different copies of them. They serve functions that are not antigen dependent. And so for that reason, we don't encode nearly as many, or the human populations do not have nearly as many allelic variations of those MHC class 1 isotypes. For MHC class 2, there's a little bit more of a mixed picture here. So for DP, DQ, and DR, which are the ones that present antigen to MHC class 2, we say they are polymorphic, not quite as polymorphic as the MHC class 1 isoforms, but polymorphic nonetheless. Specifically within HLA-DR, we do have the beta chain of HLA-DR is highly polymorphic. In contrast, though, the alpha chain of HLA-DR is relatively not polymorphic. There's only a couple versions of HLA-DR alpha. So most of the genetic diversity within HLA-DR then has to come exclusively from the beta chain. In contrast, HLA-DM and DO, which again are regulatory MHC molecules, are relatively less polymorphic. We see that for all of these, they're either only oligomorphic or, in the case of HLA-DO alpha, monomorphic. Again, we don't need a lot of different versions of them because their capacity is not antigen presentation. So we see that theme over and over again in both the MHC class 1 and the MHC class 2 genes. So I'm saying that they're highly polymorphic or not. So what does that actually mean in terms of the different numbers that we might expect? Well, these numbers, represent all of the different known, what we call allotypes, or different actual proteins that are made for each of the different genes that I mentioned. These numbers may have been updated since this was published, but they're going to be in this same general, vicinity in terms of magnitude. We're talking about allotypes here instead of alleles because not all alleles end up encoding for functional protein. Sometimes we inherit pseudogenes and we just are not able to translate them. So the allotypes are the alleles that successfully encode protein. So you can see here in the HLA locus, A, B, and C, which encode the antigen presenting MHC class 1 molecules, there are, between 1,000 and 2,000 different allotypes for these in human populations. So an incredible diversity in the possible MHC class 1 molecules that we might express. In contrast, E, F, and G, which are not involved with antigen presentation, only have very few possible allotypes. And so we just saw that in the last slide, but you can see this illustrated here actually with the numbers, with the data. If we look at the MHC class 2 molecules here, we see that most of the polymorphism is associated, again, with the antigen-presenting MHC class 2 molecules, DP alpha, beta, DQ alpha, beta, and DR alpha and beta. And what you can see actually here is that sometimes DR beta is actually even more polygenic. Some people encode even more possible DR beta genes, but nevertheless, you can see not quite as polymorphic as MHC class 1, A, B, and C, but still polymorphic nonetheless. In comparison to DM and DO, which are just regulatory HLA molecules, which have relatively fewer different possible allotypes. Okay, so we have all of these different variations, particularly in the MHC class 1 allotypes. What does this matter? What does it matter that we have so many different versions? What is actually different about them? Well, it turns out that if you look specifically on an amino acid by amino acid basis, the diversity in the different allotypes or the different alleles of MHC molecules tend to be concentrated right in the antigen binding region of the MHC. So if we take MHC class 1, for example, remember that within MHC class 1, the peptide binding groove is made up of the alpha 1 and the alpha 2 subunits. if we look at each individual amino acid in the alpha chain of MHC class 1, and we kind of graph how many different versions of that amino acid exist and all of the different alleles possible for MHC class 1, we see that the highest peaks are associated with these residues that are marked in red. And if we map those red residues onto the actual MHC class 1 molecule, we see that they are found basically exclusively within the antigen or within the peptide binding groove of the MHC class 1 molecule. So all of the different alleles, the primary difference between them are the amino acids that actually are involved with binding to antigen. So that means that these alleles are going to influence then the universe or the set of antigens that particular allele of MHC is going to be able to present to T cells ultimately when it's made into a protein. So these allelic variations, they're not random. They don't fall in any random place within the MHC molecule. They're really concentrated in the area that matters the most, which is the part that actually holds on to the antigen. We see the same thing with MHC class 2. Only the beta chain here is shown for illustration purposes, but you can sort of assume a similar thing usually for the alpha chain. But specifically within the beta chain, we see the same thing. If we take it on an amino acid by amino acid basis, we see the majority of the variation falling within those amino acids that sort of line the peptide binding groove of the MHC class 2 molecule. So a similar idea as what we saw for MHC class 1. So, okay, we've put a lot of ideas together here. MHC genes are highly polymorphic, they're polygenic, and we can encode lots of different versions of them. The reason that ultimately means that we can present a lot of different antigens is because all of these different genes and all of their different alleles typically express what we call codominant expression. So codominant genes basically means that any alleles that we have are basically expressed with roughly the same frequency, or the protein is actually made at roughly the same frequency. So, you've probably talked in some genetics class at some point about dominant and recessive alleles. For the most part, MHC alleles are never recessive. If you have it, you express it, no matter what other alleles you have. So what this means is that we can actually make then many different isoforms of MHC1 and MHC2. So here we're looking at the human MHC. So we have one chromosome that we inherited from the maternal parent, again, one chromosome that we've inherited from the paternal parent. And if we think about we exclude all of the regulatory ones. We're only looking at the ones that present antigens. So for MHC class 1, we have A, B, and C. For MHC class 2, we have DP, DQ, and DR. So starting with the MHC class 1 molecules, we inherit 1A, 1B, and 1C from the maternal parent, and another copy of each from the paternal parent, and so we end up with 6 distinct MHC class 1 alpha chains that we encode in our genomes and that we actually make into functional protein. Because we have codominant expression, none of them are repressed. We make all of them. So that means that we end up, if we call the versions that we inherit from the maternal parent with a superscript A and the ones we inherit from the paternal parent with a superscript B, then we make basically 2 copies of B. We have BA and BB, and same thing for C and A. So 6 total versions of MHC class 1 on the surface of our cell. Same thing with MHC class 2, although we have some additional diversity with MHC class 2 because remember we have the alpha and beta chains, and it turns out we can mix and match the alpha and beta chains within each of the different isoforms. So if we take DR, for example, you can see here we inherit alpha A and beta A from the maternal parent, alpha B and beta B from the paternal parent, but they can actually sort of be mix and match. So when we make our functional DR proteins, we might have alpha A with beta A, alpha B with beta B, or alpha A with beta B and alpha B with beta A. Yeah, there we go. So 4 possible combinations here. So we get the same thing with DQ, and it's not pictured here, but we would have the same thing ultimately with DP as well. They just kind of ran out of room on this figure. So if we take the numbers then, that means that the combination of polygyny and polymorphism and codominant expression, all of those things kind of mixed together, means that we can make 6 different versions of MHC class 1 proteins, and ultimately 12 different versions of MHC class 2. And so again, each of these different versions is going to bind to a different overall set of possible antigens. And so by having different versions of MHC, both for class 1 and class 2, we really cover all of our bases to make sure that we can really present a huge variety of different antigens to all of our various immunoglobulins and T cell receptors that we're making in parallel elsewhere in the body. Okay. Another idea here and that I keep saying is that not all possible versions of MHC can bind to all different types of antigens. And this idea is what we call MHC restriction. So, like I said, sometimes you just have a shape mismatch. You have a peptide antigen that has a certain shape, in which normally it could be loaded onto MHC just fine, but for whatever reason, the version of MHC that you have is just kind of the wrong shape. They just don't mesh together. And so that version of MHC is not going to be able to hold on to that antigen very well. The affinity is going to be too low. If that means that you might just need a different type of MHC with a slightly different shape, and hopefully it can grab on and then it can present the antigen. So you kind of see an example of that here in this figure from your textbook. So if we call this version of this is MHC class 1. If we call this one A, again, this is just sort of an arbitrary designation. This version of MHC, A, is bound to this antigen, which we call X. They're a good fit, and so presentation happens. This version of MHC is able to present antigen X to the T cell and activate the TCR. However, a different version of MHC expressed by the same individual, we'll call this one MHCB. It comes from a different gene and a different set of alleles. Its shape is not quite right. Maybe it can bind to X, but it doesn't hold on to it long enough, or it doesn't hold it in the right position. And so ultimately, it's never able to successfully present the antigen to the T cell, so we don't get any T cell activation. In contrast, antigen Y, its shape is not a good match for MHCA, and so Y then might be a better fit for MHCB. So you can kind of see combinatorially then of all the different possible antigens, for the most part, they end up finding at least one MHC in our bodies that they're able to successfully bind to, and then antigen presentation can successfully happen. So we call this idea of antigens only being able to be presented by certain subsets of possible MHC molecules, MHC restriction. Okay, that was the gene structure of MHC. Let's summarize. MHC diversity is an important factor in our overall repertoire of all the different possible antigens that we're able to recognize. This comes from two major factors. MHC is polygenic, meaning that it has more than one gene, so we actually encode multiple protein isoforms on those different genes. MHC is also polymorphic, so within those genes, there are a lot of possible alleles that we could inherit from our parents. So there are many variant isoforms as well. MHC, so the antigen presentation molecules of MHC, as well as a number of accessory immunologic genes, are encoded by large contiguous regions of the DNA. So in humans, that's on chromosome 6, and we call that the HLA complex. In mice, it's called the H2 complex, but they're basically analogous structures. In humans, we have 3 gene regions which encode the 3 distinct versions of the MHC1 alpha chains that we make. We call them A, B, and C. So HLA-A, HLA-B, HLA-C are MHC class 1 molecules in humans. For humans, there are also 3 gene regions that encode separate, distinct MHC class 2 alpha and beta chains. In humans, we call those DP, DQ, and DR. The possession of multiple highly variant MHC molecules is really important for expanding our antigen repertoire because, as we saw, the allelic variation that we can inherit is really concentrated specifically within the antigen binding grooves, the peptide binding grooves of the MHC molecules. And so that means that the allelic variation then contributes to the ability of all of the different isoforms that we have to hold on to a slightly different population of antigens. And because each of the different genes that we have and the different alleles that we have on each are codominantly expressed, that allows us to simultaneously express all of the different MHC isoforms that we encode in our genomes, again, overall increasing the diversity of the repertoire. Okay, so that covers our genetic discussion of, or at least on the molecular level, of MHC. So we have one more lecture in this module, and in that case, we're going to talk a little bit in more broader terms about how we inherit MHC genes, so issues of MHC inheritance, as well as HLA matching, the idea of HLA matching. So we'll talk about that next time.